Okay, folks, let's get started. Happy Wednesday. The exams are almost graded. They're not quite done yet. They should be done this evening. So the TAs have worked very hard to get them graded. I will put out an email when the exams are available for pickup, and also with instructions on where you can pick them up. So I'll put that in an email to you, and they should be available sometime tomorrow, I think. So I'll let you know about that as we get closer to it. So that'd be cool, hopefully for everybody. I don't know how the class did. They haven't told me a thing, so I'm hanging as much as you are at this point. Okay, so I want to finish up talking about membranes today, and there are some things, as we'll see, that are sort of peripheral to membranes that we will discuss because this is the logical place for us to be talking about them. And then we'll turn our attention to some fairly basic structural considerations for nucleic acids. And there we go. That's where we are. So let's see. I'm a little behind on my videos. I haven't posted yesterday's video yet. It'll go up sometime this evening. And today's will probably go up sometime tomorrow. Okay. Thought I had that set up. Okay. So last time when I finished, I was just getting ready to talk about active transport. So I want to say just a brief word about active transport, reiterate what I said at the end of last lecture, and then tell you some more about that. So if you recall, I said that membrane was a barrier to the movement of most biological molecules. And to get those biological molecules into the cell, we have to have transport proteins for most of them. And I pointed out that we can have really movement across the membrane by two distinct mechanisms. One mechanism was what I called facilitated diffusion. You also see some books call it passive transport. So it's facilitated in the sense that some protein is permitting a specific molecule to move, but it's not providing any energy for that process. So facilitated diffusion or passive transport, both of those mechanisms rely on diffusion as the only driving force to move the molecule. That contrasts with what I described later as an active transport system. And active transport systems occur when one molecule is moved against a concentration gradient. So it takes energy in order for that to happen. And so active transport systems always require energy. That energy is sometimes in the form of ATP, but not always. Okay? So that's important to keep in mind. Sometimes in the form of ATP, but not always. The example I finished with last time was the sodium-potassium ATPase. And as I mentioned before, that was a protein that you had seen earlier when we talked about covalently modified proteins. This protein is located in the membrane of virtually every cell on you, and as well as virtually every cell on the face of the Earth. And the reason for this is that this protein kicks out sodium ions and it brings in potassium ions. Okay? Both of those are against concentration gradients. That is, sodium is being moved from a lower concentration to a higher concentration. That's counter to the direction it would like to go by diffusion. And potassium is being moved from a lower concentration to a higher concentration. Potassium is being moved in, sodium is being moved out, and Bill Gates can go suck eggs or something, right? Okay. Now, the energy for this transport is ATP, and you don't have to worry about the mechanism. It is the important thing isn't how this guy accomplishes it. When we talked about covalently modified proteins, you may remember this was one that got a phosphate on it, and in the process of getting that phosphate on it, that was a step in the transport process. And from a perspective of mechanism, that's really the only thing that's relevant for this protein. The reason that virtually every cell on the face of the Earth has this and needs this is because the cell is trying to balance osmotic pressure. Okay? Osmotic pressure is a very big consideration because if you think about the concentrations of things inside your cells compared to the concentrations of things outside your cells, you've done the experiments in biology, I'm sure, where you have the membrane that is full of a solute that can't make its way out, and you put it into a solution of water, the water rushes in, the membrane starts to swell, swell, swell. Okay? The same thing will happen with a cell if you don't balance its osmotic pressure. So this protein is playing games with sodium and potassium ions in an attempt to balance the osmotic pressure. If this protein doesn't work, the cell will start to swell, 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 because remember, water moves across the membrane quite readily. So what does that mean? That means if this cell doesn't work, your cells burst. Not a good career move, okay? At least for most cells. I mean, maybe some cells, they want to burst. I don't know. But it's not a good thing. So one of the reasons that you need a continual supply of energy is to keep balancing osmotic pressure. If you don't do that, again, cells swell and burst and they die. Okay, so that is one type of active transport. And then clearly in that case, the type of active transport involved ATP as the energy source. Another type of transport does not involve ATP. Actually, the mechanism, we're not going to worry about that. Does not involve ATP as an energy source. It's something that uses what's called a secondary energy source. Okay? A secondary energy source. Now, sometimes I get into sort of discussions or arguments with students about, well, isn't ultimately ATP the energy source of all of these? And the answer is, well, I guess if you want to go back to the ultimate source, probably sunlight is the ultimate energy source of everything on the face of the earth. So we're not talking about ultimate energy sources, but we're talking about immediate energy sources here. In this case, the energy source turns out to be something that's a little hard to understand on the surface. The energy source is a gradient of ions. And the ions in this case are protons. We'll talk later about proton gradients and how they actually contain energy that can be used. All right? So for the moment, I want you to think about what's happening in the cell. Cells are very good at pumping out protons. We'll see that that is true inside the cell as well. Mitochondria, as part of their action, pump out protons. All right? So cells are good at that as well. They can kick protons out of them. And so if a cell is kicking protons out of itself, you would probably guess, A, that probably takes some energy. And the answer is yes, it does. We won't worry about that energy source at the moment because it's going to depend on the cell. All right? But if I have a cell that's kicking all these protons out, it means that the concentration of protons outside the cell is greater than the concentration of protons inside the cell. Everybody understand that? Cell's been kicking them out. Concentration of protons is greater outside than it is inside. Well, what does that mean? Okay, what it means is, what do those protons want to do? They want to go back in, right? Because diffusion says concentration gradient, higher on the outside than on the inside, so there's a pressure for these guys to come back in. The pressure for these guys to come back in is used. And it's used to carry something else in that doesn't want to come in. In this case, it's lactose. Lactose is the sugar that's found in milk. Okay? And this is a way of getting lactose into the cell. So this is called the proton lactose pump. Notice that there's no ATP on this pump. Okay? You don't see any ATP being used here. The thing that's carrying the lactose in is the protons that want, or are the protons that want to come back in. Okay? So the concentration gradient, and we say gradient, we mean a difference in concentration between the two sides. The concentration gradient of protons is actually bringing in this lactose. Now, this is called a secondary energy source because the primary energy source is the thing that's kicking the protons out in the first place. That's creating this. I like to think of this as charging the battery. You charge a battery so that you can use it later, right? The battery here is a gradient of protons. The cell is charging the battery by putting those protons outside the cell. That higher concentration wants to come in, and the cell says, okay, we'll let you come in, but you've got to bring lactose with you. If lactose is not present, the protons don't make it back in. Does that make sense? Yes, sir?
Do the protons actually, is lactose protonated and chemically bonded?
Very good question. Does lactose get protonated in the process? And the answer is no, it doesn't. So the proton is coming in apart from the lactose. Very good question. Well, again, it's the gradient. So the fact that there's a higher concentration of protons outside than inside, so diffusion works only if there's a lactose present. Otherwise, this guy is blocked.
I don't know what exactly pulls the lactose in. Does it have a...
So what's pulling the lactose in? Okay, so this actually is an energy source. What's that?
Is that like a negatively charged end that's attracted to the H1?
No, no. It actually, so we could think, when I talked yesterday about the facilitated diffusion, and I said I had a protein in the cell membrane that would allow only glucose to come in, right? The only thing that drove it in was the concentration of glucose, outside moving inside, right? Okay, so I've got a chamber here that will only allow lactose and a proton in. If I have just lactose, no good. If I have just a proton, no good. And since there's an energy associated with the proton coming in, that energy is what's bringing that lactose through. The mechanism, believe me, is a lot more complicated than we're going to go into here. But in simple terms, that's what's happening. Okay? So the concentration gradient is the energy source that's here. Now, you might say, well, if it's a diffusion process, why is it active transport? What would you say to that? Isn't it diffusion that's bringing it in? It is, but again, it's diffusing in, though, right? And I said diffusion processes were facilitated diffusion. They were passive transport processes. What's missing here? It's actually in the definition of what an active transport system is. What did I say the definition of an active transport system was? At least one molecule is moving against the concentration gradient, and lactose is moving against a concentration gradient. Okay? So that's what makes it an active transport system. This is a very, very efficient use of energy by the cell, using the energy of diffusion of protons in to bring in that lactose. If it's just diffusion by itself, if it were simply protons coming in, it would not be an active transport system because nothing would be moving against a concentration gradient. Here, something is moving against the concentration gradient, and that's the lactose. Make sense? Clear as mud? Yes, question? Oh, you're pointing at her. Okay. All right. There is a proton gradient, you bet. No, no. I said if the only thing that were moving were protons, then I would not have something moving against a concentration gradient. Okay. In this case, the protons are moving with the concentration gradient because it's greater outside than inside, right? But the thing that's moving against the concentration gradient is lactose. So that means it makes it an active transport system. For this particular thing, it has to be a proton. Okay? Most of these transport mechanisms are very, very specific for specific things. We'll see, actually we won't see, but I'll tell you later, there's a fascinating system that allows the movement of potassium ions and sodium ions differentially through nerve cells. And what's amazing is they can differentiate between the two, even though in one case, they're just basically a chamber is all it is, but in one case the sodium ions are smaller than the potassium ions, but they still can't make it through the system. It's a very cool system. If you're curious, I'll tell you more about it. Okay. Let's see. Now, there's yet another mechanism for getting things across membranes, and it's actually appropriate that it's mentioned here, although it's not usually taught at this point. And this mechanism is the way that our body takes in fat-soluble things into cells, including cholesterol. So not everything goes through protein transporters. This is a prime example of something that does not come in by a protein transporter. It's coming in by a process that we refer to as receptor-mediated endocytosis. And yes, that's a phrase that you should know. Receptor-mediated endocytosis. That last word is spelled E-N-D-O-C-Y-T-O-S-I-S. Receptor-mediated endocytosis. Now, the movement of fat in our body, as we'll talk about later, and the movement of cholesterol is rather complicated. Yes? Yeah? I'm sorry? If you want to think of it as a drag force, certainly. I'm sorry? Okay, so cells have, we'll talk about pumps later, but cells have ways of transporting things out to accomplish something. So, and you can have, for example, an ATP-driven system that kicks the protons out